Hey there, and welcome. Today we're pulling on our lab coats and grabbing our magnifying glasses to become chemical detectives. We're going to find out how scientists can look at pretty much anything and tell you exactly what's inside.
I mean, think about it for a second. It's a really important question, right? When you take a pill, you trust it's the right medicine. When you drink some water, you trust it's clean. But how does anyone actually know for sure?
Well, here's the thing. Almost everything out there is a really complex mix of different stuff. A single drop of water from a river, or even a tiny sample of your own blood. It can have thousands of different chemical compounds swirling around in it. So for scientists, the real trick is figuring out how to find that one specific thing they're looking for, that one needle in a massive, massive haystack.
To pull this off, scientists have this incredible one-two punch of techniques. You can think of them as the ultimate chemical detective kit. You've got one tool for sorting all the evidence and another tool for figuring out what that evidence is.
Okay, first up, we have chromatography. This is the separator. Its whole job is to take a messy, jumbled up mixture and neatly sort it into all of its individual parts. Here's a cool way to think about it. Imagine a marathon. All the runners start at once, right? Well, chromatography is like the race course. Based on their own unique chemistry, the different molecules or runners move at different speeds, and eventually they cross the finish line one by one, all perfectly separated. So once all our runners are separated, we still need to know who they are, and that is where mass spectrometry comes in. This is our identifier. It takes each of those purified little components and basically weighs it to figure out its molecular mass with unbelievable accuracy. And that specific weight, it's like a chemical fingerprint that lets scientists say, aha, that's caffeine or that's ibuprofen. So how in the world does this molecular scale actually work? Let's kind of follow a sample as it zips through the machine. You see there on the left, it gets injected. Then it's turned into a gas. And then, zap, it gets hit with an electron beam, which gives it an electrical charge. And this whole five-step process is the key. Once those molecules are charged up, they get shot at high speed into a super powerful magnetic field. Now this is where the real magic happens. Imagine the wind trying to push a bowling ball versus a ping pong ball. The magnet pushes the lighter molecules way more than the heavier ones, so a detector at the very end can measure exactly how much each molecule was bent, and from that, it can calculate its precise mass, and boom, you know what it is. Now look, these two tools are pretty amazing all by themselves, but the real game changer is when you hook them up together. In the chemistry world, they actually have a name for this. It's called hyphenation. Yeah, just like this research paper says, hyphenation literally links the two machines together. So as the chromatography machine is separating the mixture piece by piece, it shoots each one straight into the mass spectrometer to be identified right away. You get the best of both worlds, all in one go. And honestly, this slide just sums it all up perfectly. Chromatography does the separating. It's like sorting all your puzzle pieces by color and shape. Then mass spectrometry comes in and tells you exactly what each of those little pieces is. And together, they let you see the whole picture. They solve the puzzle. Okay, so this dynamic duo actually comes in two main flavors, and each one is a specialist for different kinds of mysteries. You can think of them as two totally different detective squads. One works with liquids, and the other works with gases. So first, let's look at LCMS. That stands for Liquid Chromatography Mass Spectrometry. With this setup, your sample is dissolved in a liquid. You can see on the left of the picture, you've got the solvent bottles and the machine that does the separating. The separated parts then flow right into the mass spectrometer on the right, and you get an ID almost instantly. The other detective squad is GCMS, or Gas Chromatography Mass Spectroscopy. This one is for things that you can heat up and turn into a vapor. The sample gets injected and heated, and the gas then travels through that super long coiled tube you see in the middle. That is the race course where the separation happens. And from there, just like before, the separated molecules zip right into the mass spectrometer to be analyzed. So how do scientists know which detective squad to call? Well, this table kind of breaks it down. The single biggest difference is how tough the molecule is. See, LCMS is for the delicate stuff, molecules that would just fall apart if you heated them up. That makes it a huge deal in drug development. GCMS, on the other hand, is for the tougher compounds that can handle being vaporized, which is exactly why it's a favorite in forensics. So here's the easiest way to remember it. If your molecule is fragile, kind of delicate, and can't take the heat, you're going to use LCMS. But if it's tough enough to be turned into a gas without breaking down, then GCMS is the tool for the job. Simple as that. You know,
This whole distinction based on heat actually brings up a really interesting point. In science, the little details, the context, it's everything. And to show you what I mean, let's look at this really fascinating little puzzle about how temperature affects how gases behave.
So, a teacher once suggested this idea about how a gas escapes through a tiny pinhole. And the logic kind of makes sense at first. You heat up the gas, the molecules start vibrating and bouncing around like crazy, which would make it harder for them to neatly line up and get through the hole. So, higher temperature means a slower escape.
It sounds pretty logical, doesn't it? But is that really the whole story? You know, when it comes to the laws of physics, our intuition can sometimes trip us up. Because the real answer is, it depends. It all comes down to the conditions of the experiment. If you keep the density of the gas the same, then yeah, a higher temperature actually makes the gas escape faster. But if you keep the pressure the same, then a higher temperature makes it escape slower. It just goes to show you why in a lab, for a technique like GCMS, being able to control every single variable, pressure, temperature, flow rate, is absolutely critical to getting a result you can trust. Context is king.
And, understanding all these little technical details is what makes these tools so incredibly powerful. So let's zoom back out from the lab and talk about why all this stuff really, really matters in our day-to-day lives.
The truth is, the applications are just everywhere. These techniques are absolutely essential for making sure the drugs we take are safe and effective. They're used in forensics to find poisons or tiny traces of evidence. They monitor our water for pollution, they check our food to make sure it's pure, they catch athletes who are cheating, and they even screen for explosives at the airport. So every single day, in countless labs all across the globe, these chemical detectives are silently at work, analyzing, identifying, and measuring. They really are the invisible systems that help guarantee the safety and the quality of so much of our modern world.
It really makes you wonder, doesn't it? These tools were built to solve the big chemical mysteries of their time. But as our world gets even more complex, as we face new challenges like brand new viruses or incredibly subtle pollutants, what are the next mysteries going to be? And what kind of tools are we going to have to invent to solve them?